Great. And welcome everyone. Thank you so much for joining us, and some of you repeat attendees to our session, so we're glad to have you back. Welcome. My name is Chris Herman. I'm going to be the session moderator for today. Heather and Brian are also going to support behind the scenes. So they'll be entering some things in the chat and answering some questions if there are 3D molecular design related chat questions. And Chris Chou will be our presenter for the next 75 minutes. Like I said, please feel free to use chat to communicate during the session. And also, I think if you have questions, it would be okay to unmute yourself and ask. But I now have the privilege of introducing 3D Molecular Design's model teacher, Chris Chou. Chris holds a bachelor's degree in bioengineering and a master's in curriculum and instruction from the University of Illinois at Urbana-Champaign. She's been a science educator for 20 years and enjoys facilitating workshops for science teachers in her district and at national conferences. She received the 2014 Spotlight Award for Excellence in Teaching from the St. Vrain Valley Education Foundation and the 2016 Outstanding Biology Teacher Award in Colorado from NABT. She currently teaches AP Biology and Biotechnology and coordinates the Medical and Bioscience Academy program at Longmont High School in Colorado. So let's now begin the program and take it away, Chris.
All right, thank you. And thank you for all you participants who have joined us today, especially to those of you who were willing to come back and hear me share about some teaching tools again. We were, just to let you know, we were taking some bets on how many participants we thought would show up today based on this morning's attendees. And you guys have exceeded our expectations, maybe doubled or tripled. So thank you for being here with us. Today, we're going to use the 3D Molecular Designs student kit, and I'm going to show you also how I use the classroom kit to model chromosome structure and anatomy. We're going to compare and contrast mitosis with meiosis, model mechanisms that contribute to genetic variation, and also throw in Punnett squares as well. So definitely something to be able to help freshman biology students to understand in a visual format how the independent assortment happens with alleles, and to be able to have offspring, projections. So from these Punnett squares. All right, so let's go ahead to the next slide. And I think we've already had those who were here a little bit earlier talk about where they live and what they teach. So if you haven't typed that in the chat yet, feel free to do that. But I thought I'd kind of share a little background of why I'm really passionate about using these 3D molecular designs kits. First of all, these are photos from my classroom. These are my students from 2 years ago, pre-COVID. And you can see all the variety of 3D molecular design kits I use, and even they're genuine smiles on their faces, actually. And my students really like using kinesthetic materials, whether it's these kits or I use Play-Doh and things like pipe cleaners and markers on these black tables you see, for students to be able to model in visual ways or physical ways their understanding of scientific concepts. So one fun story is these girls in the middle who you see with the DNA, they actually are modeling replication right there. And they had interacted with the kits in class, and then we kind of ran out of time. And they asked me if they could come in at lunch to use the kits, and not just one kit, because I put one kit per table. They're like, we need 2 kits because we want to show both replication forks. And so they got out 2 kits and did that on the table on their own time because they loved using these materials so much. But I got into figuring out how to use these kits from attending workshops, both at places like science teacher conferences with that 3D molecular designs put on, and summer workshops as well. So I know that I think Heather can post some information about potentially upcoming summer workshops in Milwaukee, where they're located. And it's a great city, and I love being able to travel and get out of Colorado and to go there. So want to plug that for all teachers here. Okay, so as we move on, this is pictures of the classroom kit, and you can see that I have some trays set up with these foam chromosome, and you can see the spindle fibers set up. Those are the yellow things right there. And so I usually have students in pods or groups like that and using a kit per group. But today we're going to use the student modeling kit. And if you have pre-registered for this conference or this session, hopefully you have this kit in front of you. If you didn't have time to assemble it, no worries. But there is a little bit of what I like to call arts and crafts stuff, and students love that. Anytime I'm like, get out scissors and glue and tape, they're all in. So you'll need to do a little bit of assembly. So cutting some graphics out and then taping some things together like this really large Punnett square. And if you don't have this kit, because you just kind of were browsing the options and popped on in, you can sign up to receive a student modeling pack. So Heather's put that link in the chat, and you can click on that and get your own kit. So last year during, a lot of virtual teaching at the beginning of the year, we were 100% virtual. And so I knew that was coming along, and I ordered multiple kits for students to have at home. And there was a whole drive-through line. Kids came in, picked up all their supplies and took them home. And they really appreciated having these kits at home in a year where we couldn't do a ton of hands-on labs. So this was a way for them to be able to interact with materials at home. And some students returned them to me at the end of the year, and others were like, could I keep mine? I really liked using them. So that was kind of fun. So this year we're back fully in person with masks. And so we get to do labs again. So I'm back to using the classroom kits, but I do want to show how you can utilize these individual student kits, especially if you're still virtual, or maybe have some students who are, being quarantined or isolated. That way they can participate too. Okay, so what I want to ask participants who have these kits to do is go ahead and take your materials and assemble the chromosomes that are shown here. And this is like a portion of it. Their foam parts on the end are sort of the larger pieces, those reds and blues. And then there's the rainbow color, red, blue, yellow, green ones. Those are the nucleotides, and you can fit them into this scaling graphic. And we're going to talk about what's there. So I'm just going to pause right here. So as if folks are assembling objects, they can do that. And I just want to say that the nucleotides in the middle, if you're trying to locate, all the different colors, it doesn't have to be any specific sequence right there. It's just a demonstration of that those are DNA nucleotides that are going in the middle there. So you can assemble that, put that together. And here, I'm going to just show on my camera, here's my 2 chromosomes here that I've assembled. And so now we're going to go through talking about the organization of eukaryotic chromosomes. So you notice these are in linear fashion. So this is a linear chromosome, which are eukaryotic genetic material. And so looking at this diagram and then comparing your model to this diagram, can you identify the different parts as I go through them? So at the very top, you see the DNA double helix. You can see how it's twisted in helical form, and the nucleotides are paired up on the middle part of that DNA ladder there. And then as the DNA is coiled up and organized, it's wrapped around histone proteins. You can see the little cursor pointing there. And can you identify that on the little paper piece of this scaling graphic? And then as we kind of zoom out and take it a larger picture or image of chromosomes or DNA, you can see that there's further condensing or coiling of the chromatin fiber of those little nodules. Those are nucleosomes. And then the further condensing of DNA as DNA is getting ready to be separated in mitosis or meiosis. And so at the very bottom, you can see the sort of straight X structure of the duplicated chromosome. It's really interesting being able to use this kit because it can reveal student misconceptions about their mental image of what a chromosome is and what that X is. In fact, some of my students are like, they look like flies, because you might have seen them on a karyotype. And a lot of teachers have these same misconceptions too, that the X is what chromosomes look like. And so we're going to talk about in a little bit, really helping students to identify and make sense of these different words that start with C. So that can be really confusing for students. So I like this scaling graphic because it can show DNA at different levels, from the molecular level of the nucleotides, and as you zoom out to the nucleosomes and then the condensed chromosome. And so that's really helpful. So the comparison of your student modeling kit is on the right. You get a handful of nucleotides, and you can build two pairs of chromosomes. And then the chromosome connections kit is the classroom set, and you can see on the left, and all the nucleotides, either you can put them in or as the blue ones all together and the red ones all together. And that's helpful, as we'll show in a little bit, for showing crossing over happening between maternal and paternal chromosomes. All right. Okay, so now next task is we're just going to focus on the chromosome level, the large chunky structures there. And so you'll need to take off from your scaling graphic these chromosome pieces and use the rest of the kit there to assemble these 4 chromosomes as shown. And you're going to notice you will have some leftover pieces, and that's for a purpose. And it really doesn't matter where you put the little indentation part, but just so students can see that that middle part is the centromere, and centromeres can be right in the middle. It can be slightly off center. So it's actually not a bad thing if you just tell students, hey, construct it, take this many and make these chromosomes. Okay. All right, so let's look at the different parts. I usually walk my students through as they have their foam pieces in front of them. Even with my AP students, I have them take their pointer finger and actually point to the parts, because I want them to show me that they understand where the parts of the chromosome are. So the ends are the telomeres or telomeres. Blues are going to represent paternal chromosomes, and the reds are representing maternal chromosomes. And of course, you've got 2 of each type of or size of chromosome. Those are the homologous chromosomes. So I, at this point, have students construct them. We go through this slide. I have them point to the parts, and then I have them stop and share with each other. So it's a good way to get students talking about science content in constructive ways. We know they all like to talk about what's happening on Snapchat and TikTok. This is a way for them to have science conversations in the classroom. And then, of course, the centromere, that's the region of the chromosome where there's more condensing there. And this is where also the location of where these homologous chromosomes will pair together and stick together when we are getting ready to divide them out. Okay, so this representation is the human karyogram. And what that is a visual showing the staining pattern of each type of chromosome in a human cell. They start off with 1, which is the largest or longest one, and then all the way down to the sex chromosomes, the X and Y. And then this graphic is showing how each of those karyogram representations is representing 1 copy of a chromosome, but because we are diploid, humans have 2 copies of every single chromosome. And then of course, the sex chromosomes at the end, you have 2 copies of X, or 1 X and 1 Y, or in unusual genetic conditions, you can have students look at karyotypes and try to figure out what would be the biological sex of this individual based on their karyotype. So let's go on to an actual picture of a karyotype. You can see here that we've got a picture of actual chromosomes within a cell, and they are at the stage of where they've been replicated already. This is where the photo is taken. So each long line that you're seeing there is actually a replicated chromosome. So I'm on Facebook in a biology teachers group. I'm just going to take my pointer here. I hope folks can see it. Teachers were asking are these the replicated chromosomes, are these identical? And the strand on the left and the strand on the right, they are not identical. Where the identical parts come in is you have to kind of zoom in very closely. This one, what looks like one rod, is actually kind of a squished X. So a lot of students think, oh, these are the two that come together to form that X. No, the X is actually one of these rods, and the two like arms of this are actually together. So if you look super close at some of these, you'll see that each of these rods is a replicated chromosome. So what you're looking at there is 4 copies, if you will, of those particular chromosomes. Okay, so super important to show students that karyotype, and if you can, zoom in, because they get confused about what they're seeing with these karyotypes. So the karyotypes are made when they have cells grown in a petri dish in the lab, and then they're going to arrest their development so that they can take a snapshot of these cells as they're going through division through mitosis. And oh, I see. Hang on just a second. I need to be able to, oh, well, never mind. I'll just have Chris point. I was like, my handlers are helping me to figure out the screen here. Okay, so getting a picture of the karyotype, it's important for students to understand the cell cycle, that the cell goes through periods of where there's just normal cell growth and development at G1 stage, and then getting ready to divide at the DNA synthesis or S phase of the cell cycle. And then the cell has checkpoints along the way. And then G2 is still part of interphase, that in between cell division stage where everything's sort of checked. Are chromosomes replicated correctly? Are there mutations? Do we have enough of proteins that we need for cell division to be able to move forward? And then once the cell goes into the mitotic phase, during metaphase is when development is arrested to be able to take a picture of those chromosomes that have been condensed, and it's easiest to see them at that point. So it's helpful to have students know, where these pictures are taken, where they're coming from. All right, and so let's do that right now. Let's do that mitosis, but before we can do that, we got to go through the S phase of interphase, and that is where DNA replication happens. So the chromosomes are going to be replicated there, but we can't see them in the cell visibly yet. They don't appear condensed yet until the M phase of mitotic phase. But let's go ahead and do this so students can see where they're getting replicated and what happens. So now you want to take all the rest of your foam parts and make an identical copy of each chromosome that you made earlier. And these are going to be identical copies, so they should have the same number of segments. The centromere should be in the same location. And you want to go ahead and keep them together just like you're seeing in these graphics, and not have little stems stray or whatnot. They kind of need to stay together. So each of these chromosomes that you are seeing are now replicated. So I like to say they're replicated chromosomes or duplicated chromosomes, and they're made of 2 identical sister chromatids. So this is where students get really confused with all the terminology in biology, where you've got centromere, chromosome, chromatid, and chromatin. And so it's important if, for students maybe to say out loud what each item is and what, and actually point to different parts so that they can identify these different terms that are being used. So each of these structures is a chromosome, but it is a chromosome replicated. And so I will actually have students in the classroom just all stand up. I do a lot of kinesthetic stuff in my class. Have them stand up and go, this is a chromosome. And actually it helps, I'm stripy today, so I can be like a stained chromosome here. And then, so this is a chromosome, this is a chromosome replicated, and have them do like a jumping jacks kind of thing where their arms and legs are out. So you can see that there's like, each half is an identical copy of the other half. So you know that you are a chromosome replicated. So those X's that you often see in the pictures in the textbook is actually one whole structure that you're seeing here that's called a chromosome. So that terminology is also, so you got chromosomes, chromosome replicated, both are called chromosomes. Okay, so let's move on and let's go ahead and go through our division of these replicated chromosomes. And of course, that's mitosis. So this is often what students see in that textbook of the what looks like flies. Now you might not be able to unsee that anymore. They look like kind of little flies in the cell with these strands of yellow coming out, and that's the spindle fibers. And so you're gonna go through prophase, metaphase, anaphase, and telophase. And so in this kit, what's helpful to know is you're going to have your chromosomes, okay? And your kit doesn't have the spindle fibers or microtubule spindle fibers that come with the kit for the student modeling kit. So students can just use like string and then kind of lasso it around. You got one for each of these sister chromatids to demonstrate spindle fibers attaching to them. So that's how you can use the student kits. You can also have students just lay them down on the table and use table markers and draw them out. But if you buy the classroom kit, it's really handy. You actually get the foam parts with the spindle fibers of this rod. And then you have, this is the protein complex, the kinetochore region that attaches to your centromere of your chromosomes. So then on a table, students can show during prophase, these chromosomes condense and become visible, and the spindle fibers attach to each chromatid. And then they can actually use these spindle fibers to pull and move chromosomes to the two poles of the cell. So let's go ahead and use our student kit now and show the attachment of chromosomes to the spindle fibers, the arrangement of them at the metaphase plate, anaphase, and then telophase. Okay, and so if we go to that slide, this is what you can see, and have students do it on a table, or if they're at home on their desk, you might want to ask them to spread out their materials on the desk so that they have lots of room for this. And in a classroom, you can do this on a lab table or whatnot. And also, add in markers or other materials so that students can demonstrate what happens with the nuclear envelope, how it will disappear, degrade, and then reappear during telophase. So hopefully you've shown each of these phases on your desk at home. And then the next slide is going to show you an animation of that happening. Okay, so this is 3 pairs of chromosomes. So this is the classroom kit, and you can see the yellow foam spindle fibers, and then they're pulled apart, and then you have, there you go. So, okay. So at this point, it's really helpful to circulate around the room and ask students some probing questions to see, do they really understand the process of mitosis, that you're starting from one cell and making 2 copies of 2 identical copies or clones of that parent cell. And then really important to ask them in terms of numbering, how many chromosomes were in the parent cell, how many chromosomes replicated, how many chromatids, and then how many chromosomes are in the daughter cell. And so using these terms are really important for students to be able to articulate their understanding of, because biology, as we all know, is like another foreign language. So that's really helpful. Okay, so now they've unlocked the basic version of cell division, and we would move to meiosis. So this is our double division happening here. And you can see that we're going from 1 parent cell to 4 very unique daughter cells. And if you do the counting of chromosomes, we're going from 2 sets of every chromosome type to just 1 copy of each type of chromosome in there, from diploid to haploid. So we're going to show that with our foam models. Now, this part is where I'd used a lot of different kits. I've done pipe cleaners, I've done string. I did even playing cards one time because I wanted students to model, crossing over and homologous pairing and all that. And I found this kit to be the most helpful in showing crossing over. So this is where the power of using these foam kits comes in. Okay, so let's go ahead and go together and model through this. So you're going to take your 4 original chromosomes in G1 phase of the cell cycle. That's the first picture. And then you already have your pre-made chromosomes, which will speed up DNA replication in S phase. So now you have 2 identical copies of each chromosome there. And then now we're going to get to the fun part. So you've got we are going to go into meiosis, and the special part of that is prophase 1, where we get to do some crossing over. So this graphic right here is showing you how crossing over happens. And really the purpose of it is to be able to align these homologous chromosomes so they're connected together and stay together all the way into anaphase 1, so that they're paired accurately and the correct chromosomes are divided out, so we don't have inaccurate numbers of chromosomes in our resulting daughter cells. So that's really the purpose of meiosis, or purpose of crossing over, is to be able to join these homologous chromosomes together. So going deeper, maybe at the college level or AP level, you could also talk about these places where they join together, holiday junctions, how that crossing over happens. That's beyond the scope of AP. That's kind of what I like to say to my students. But I like to show them this to say, hey, it's complicated. Life is complicated. Let's go to sort of the basic foam version of this. Okay, so this animation is showing you with the classroom kit, the red nucleotides representing the maternal chromosome, and then the blue nucleotides representing paternal chromosome DNA sequence. And you can actually go through and model these holiday junctions, the breaking of the chromosome strands, and the reconstruction of them using additional nucleotides. And then so that's crossing over at the nucleotide level. So this is really what's happening. So it's literally joining and then swapping of parts. And so you have this swapping. So let's go ahead and model that with these foam parts that we have here. So synapsis is when the homologous chromosomes come together. So you form tetrads where all the like chromosomes will find each other, the maternal and paternal homologs. And so you end up having 4 sister chromatids all together. And then crossing over. So there's a couple of ways that students can do this. So I am showing you in this picture how you can line up 2 pairs of chromosomes side by side, and then you can easily see how the maternal and paternal arms are swapping pieces right there. So there's a literal removal of, so I'm holding this, I'll do this on the camera here. Okay, so there's the synapsis where they're lining up. And then you can actually have them remove foam parts, okay, and then swap them. And so this right here is the power of the kit, because you can actually, without destroying, what did I use before? I used pipe cleaners and I used socks. So there's this kit where they use these socks and you would pair them, but you couldn't like cut the socks and move them around and re-tape them or knit them back together. But these foam kits are really easy to do that. So you would do this crossing over and swapping of pieces, and it's pretty violent. As I tell students, it's like you're cutting and snipping DNA and then moving them around. And so the crossing over will happen between 1 to 3 places. And so there's 1 to 3 crossover events per chromosome. So now this is showing you in a 2D form. But what can be really helpful for students is to understand this is happening inside a cell, which is a 3D space. And so instead of these chromosomes sort of lined up next to each other, as textbooks can only show, you can actually have them just back to back, as you see in my camera, and have them lined up and then just do the cutting and the swapping that happens right here. So students can understand that inside a cell, this is 3D space, and these molecules are just lined up and moving around, and this is where how the crossing over can happen. So I think this is a good usefulness of the kit to help kids conceptualize from 2D pictures to 3D space. All right, so let's go ahead and take a look at have you model that? And so this is the graphic again. And the next slide will show moving your 2 pairs of chromosomes to do synapsis, crossing over. And in this picture, it's only showing 1 pair of chromosomes crossing over there just to make it simpler. And then so you can move them through all the different phases of meiosis. So meiosis 1, and then the second division, meiosis 2. And you can see now when you end up with, at the very end, after telophase 2 or telophase 2, you have 4 different daughter cells. And when you count the number of chromosomes in there, students can see we started with 4 chromosomes and now you've got 2. So they're now haploid. And not only that, but the chromosome molecules themselves are not in the original form. And so some of them have that crossing over. So you have this mixture of red and blue parts. And then also the fact that some cells could have different maternal or paternal chromosomes based on that assortment, the independent assortment of these chromosomes. Okay, so let's take a look at the classroom version kit, this animation. All right, so one more time. There is the crossing over, metaphase 1, anaphase 1, telophase 1. Here's prophase 2, metaphase, anaphase, and telophase 2. So that's kind of fast there. You're probably going to want to take it much slower in the classroom. And this is really revealing when you watch students move the chromosomes around on a table, because that's where it will show whether they understand which chromosomes stay together, that the sister chromatids of each chromosome replicated stays together, and then which sizes or how they line up in pairs along the metaphase plate or equatorial plate in metaphase 1, and that lining up that's where that independent assortment comes from. And then as they separate out. So usually the first time I have students doing the end of, all of these steps completely wrong. And I can see that right away as I see what the resultant cells are. And so this is a really powerful teaching tool, especially for teaching meiosis. It's pretty difficult for students to explain that. They just see the pictures in the textbook, and having these chromosomes in hand to move things around is really helpful for them and for the teacher to see what students understand. So again, at the end of all this, you can ask your students, how did the daughter cells compare to the original parent cell? The number of chromosomes, keeping track. At the AP level, we talk about whether these cells are diploid or haploid and what that means. And then you also have to do some comparison. How are mitosis and meiosis similar? How are they different? Maybe where are the steps or stages at these similarities and differences? So good ways to have students talk about science and converse and show. This kit also helps students to see what are the ways that DNA can be shuffled or mixed up to be able to produce such incredible diversity within our species. And so you can ask students about the source of sources of genetic variation, of course, with the crossing over happening at metaphase 1, and then the separation of homologous chromosomes with anaphase 1, law of independent assortment. And then finally at the end, when sperm and egg come together in fertilization, that's another place where you have some shuffling of DNA and different mixtures and varieties of genes that come into play. So, all right, so that's the basic uses of this kit. And what I always like to do is bring in real world examples for my students as well. For some of them, it's to help them understand what's going on with their own bodies, or cases of, for example, genetic diseases or health conditions for them. And so I like to have them play a genetic counselor role for this particular topic. And so let's meet a fictional family. They're the Smith family. Can't get more sort of generic than that, Smith, or you could use whatever last name you want to represent your student population, where they have representation. Okay, so let's meet the Smiths. And they are a young couple who come in for some genetic counseling and have a unusual genetic condition that runs in their family, and they want to know if they have kids, what will be the chances that their kids might inherit this condition. So let's talk about the gene involved here that may have a genetic mutation. And so let's talk about the HPV gene. This is the beta globin gene that produces the protein that's the beta globin, which is one of the subunits in the oxygen carrying protein called hemoglobin. So you can see here that hemoglobin is found in red blood cells, and it's constructed of 4 subunits, 2 alpha chains and 2 beta chains. And within them, there is a heme group. So that's where that iron comes in, which helps to carry those oxygen atoms or oxygen molecules for these red blood cells. So the main job is to carry oxygen. And so because of these nice roundy shapes of these proteins, you get these red blood cells that are nice roundy shapes. And of course, the reason it's not a spherical cell, it's because after these red blood cells mature, the nucleus is ejected. So what you get is this nice round biconcave shape of the red blood cells. All right, so what can happen with this HPV gene? So this genetic counselor will tell this couple about how this gene's located on chromosome 11. So it's important for students to understand all these genes we talk about. There is a location for them. There is an address where they usually reside. And this one is on chromosome 11. And on this chromosome, it's on this arm of the chromosome. So there's a specific location, and it's been identified through, staining patterns and where this gene might be tagged or located. And when we zoom in, these genes have a certain sequence, and people may inherit different versions of the sequence. And these are all called alleles. And so if we zoom into one particular location or locus along this gene, we see that most folks have a version which is their DNA sequence is on the left. You see the GAG on one strand and the GAG and then CTC on the other, on that one, on the complementary strand. So we call that the normal beta globin protein version at that location on the gene. On the other side, on the right side, you see where one of the letters or nucleotides has been changed. So instead of that A-T pairing, you now have a TA pairing in the middle. So it just seems like it's very little of a change. Like, will it make a difference at all? And so let's take a look at the bigger picture. Well, DNA codes for making RNA. And of course, RNA is a sequence used to be able to figure out how we're going to construct our proteins and put together our amino acids. So in that sequence, the original version of the beta globin gene at that location would normally code for a GAG in RNA, which will translate to a glutamic acid in our amino acid sequence, in our protein primary sequence. Now, if you have that slight substitution what's going to happen is instead of a GAG for RNA, you get a GUG. And when the ribosome is reading that strand, instead of a glutamic acid put in, you will have valine put in instead, because GUG codes for valine. And so you got this little bit of a change. So it's a missense mutation because you have an amino acid, but it's a different one than was originally coded for. So will that make a difference? Well, let's take a look. These proteins rely on the primary sequence of the amino acids to be able to then fold up into a 3D shape. And having that glutamic acid there at that location means that the protein, the beta globin protein, will be able to fold up into a nice like round globular shape. And they interact with these other subunits of hemoglobin of like alpha and beta globin. And so that, that amino acid side chain glutamic acid, that is hydrophilic. And what that means is as the protein is folding, that particular chemical group is attracted to water and tends to face the outside of the protein right there on the left. Okay. Now, what happens when we change that one amino acid to valine? Well, it turns out that valine doesn't have the same property. It is hydrophobic. And so as the protein is folding up into its 3D shape, that valine is problematic because what it creates is kind of a sticky spot on that part of the beta globin protein, causing other beta globin proteins, which also have that same abnormality, to start to clump together, and they kind of stick together. And in fact, as they stick together, there's this whole long chain of them that clump together, and you can see that on the right side of the screen. And so in biology, one of the mantras that I tell my students all the time is that structure dictates function. And that changes in structure lead to changes in protein function, or any function, whether it's we're talking about organelles or molecules or enzymes. And so in AP biology, I actually, in addition to talking about protein folding and looking at genetic mutations, such as with the beta globin gene, I also bring out, I've purchased a map, the human beta globin gene map. And for those of you who are teaching college level or AP biology, this is at a deeper level. I have my students take a look at the sequence of the beta globin gene. And also the fun thing about this map is, besides that one spot I mentioned where there's a variant there, there's other locations along the same gene that can have other mutations that occur, which leads to versions of sickle cell anemia that are more severe or less severe depending on the location of those mutations. And then students can also take a look at ideas such as introns versus exons, reading frames for the ribosomes, codons, and then where those mutation sites are located. So that's in the teacher version of that strip. And then students actually take a look at this big long strip printed out, and I have it taped on the wall or on their desks to be able to look at the sequence and be able to see how the DNA sequence affects the protein structure later on. Okay, so that's a little side note for there. As we move on, let's go back to those original variants, the zoomed in spots we looked at. So when we talk about genetic mutations and inheritance, it's important to know that, that where they come from, first of all, and then those loci, the locations of where the mutations occur on the chromosomes somewhere. And so that's going to be affecting when you do meiosis, and assorting chromosomes and making sperm and eggs with these chromosomes, those mutations get passed on to those cells. And so going back to the DNA, we constructed hemoglobin A and hemoglobin S and at that location. And so students can take a look at different combinations you could inherit. So if you could have 2 copies of normal hemoglobin, normal beta globin, you could have 1 copy of a normal version, 1 copy of the abnormal version, or 2 copies of that abnormal version. And so this is where we get those genotypes from, and assigning these letters to represent these different alleles or variants of this gene. So if you inherit 2 copies of the normal beta globin, you would have a normal phenotype. Your proteins would be shaping just fine, and they would carry oxygen well. No, you say you have a normal phenotype. If you have 1 normal copy and 1 abnormal copy of the beta globin gene, you end up having what's called sickle cell trait. So you would be a carrier, and on the outside, you would appear to have a normal phenotype. Unusual things usually would not show up, and you wouldn't even know that you were a carrier for an abnormal copy of this gene, unless you encountered some unusual situations, such as going to higher altitude, or some athletes, may discover as they have trouble with, oxygen levels or breathing, or, some kind of circulatory issues. They may get tested and find out they're actually carriers for sickle cell anemia. And of course, if you get 2 copies of the abnormal beta globin gene, you end up getting sickle cell disease. And so that's how the genotypes show up and what they mean. And in terms of what that means physiologically, having the normal hemoglobin molecules allow your cells to be in that nice roundy shape. And if you have the abnormal hemoglobin molecules, they're going to stick together, form all those rods that we saw earlier because of the sticky spots. And that means these cells are misshapen and forms the traditional sickle shape there. And as these red blood cells are being transported around the body, you've got the red blood cells will go through the smallest vessels, which are capillaries. They have to go through in single file line. So what happens if they're misshapen? And so you get the picture down below where these little cells get clogged up. There can be blood clots forming in a variety of organs and whatnot. And this is what's causing that sickle cell anemia, is some things showing up symptom-wise with these little cells. Okay, so going back to our couple, understanding the origin of this HPV gene, different variants that you can inherit, and then translating that into okay, these are the alleles, and we're going to assign letters, and let's take a look at what could happen. And so the Smiths, turns out, they get genetic testing done and find out they are actually both carriers of sickle cell disease. And so they have the sickle cell trait. And so they both each have 1 normal copy and then 1 abnormal copy of hemoglobin. And so now kids can take a look at and figure out what kind of eggs and sperm what can they carry? And then if these eggs and sperm combine together, what potentially would be the genotype of their offspring. And so this is where this graphic comes into play. So in your student kit, you'll get a large cardstock copy of this Punnett square. It's really large, and it's meant to be able to actually form the sequences of those codons right there of those triplets to show the 2 variants that Mr. and Mrs. Smith could that they have. And then you can move these particular nucleotides to have different combinations. So you can see whether the offspring can inherit 2 normal copies. And then if we click on it, I think it shows the other ones that pop up. Okay, and so you can see the different combinations of these alleles that can combine together for the students, or for the offspring right there. Okay. And all right. And so as they look at the potential projections of, what could those offspring inherit, what are the chances the Smiths will have a child affected with sickle cell disease? And so students would take a look at their Punnett squares and identify that, this disease is inherited as an autosomal recessive genetic disorder, where if you at least have 1 normal copy, it kind of overrides the negative effects of that abnormal copy. And so if you, by chance, get 2 copies of the abnormal beta globin gene, you would inherit that condition. And so 1 out of the 4 children with that little h genotype would be affected by sickle cell disease. And so that the chances then, as they do the math there, 1 out of the 4 would be 25%. So it'd be 25% chance of inheriting that condition. So in talking about what happens now, this is a really great classroom discussion of, as they play this role, or maybe even like as the parents, what kind of options do they have? And so in really educating parents and families or couples about what can happen with their children, and then guide them to resources and support for families who have sickle cell disease, maybe in their genetic background. So that's a really good conversation to have. And also there's some good news too, because as you've probably heard in the news, one option for certain individuals who are afflicted with sickle cell disease is CRISPR-Cas9 gene therapy. And one thing that's great about this particular single gene disease is because it affects these red blood cells, they can actually remove cells from the patient who's afflicted, genetically alter the cells, and then replace them back. So for those of you who are part of my workshop yesterday when I talked about cystic fibrosis, that is a very difficult disease to cure or treat using CRISPR-Cas9, because this problem shows up in every single cell all across the body. But sickle cell disease mainly affects these blood cells. So if you can repair them or get functioning genes on maybe some of the other hemoglobin related genes, then that could potentially mitigate, the severeness of these diseases. So kind of cool. You should look that up if you haven't read that. So it turns out, besides kind of sort of giving you a foreshadow, besides beta globin, there's other versions of the globin gene family. There's alpha globin, there's adult and fetal versions. And so the gene therapy patients who've undergone treatment have actually, I think they've turned on fetal hemoglobin for those patients, and which means it's restored some function of oxygen carrying ability. And so that's been enough to keep them out of the hospital and help them to live, relatively, pain-free lives. So anyways, that's exciting. Another note, maybe that's part of a CRISPR workshop there. But here's some of the standards that are covered through what we've talked about today. And I hope that you've seen the usefulness of these modeling packs, whether it's the student version or the classroom version. This one, I think, is a really powerful tool to reveal student understanding and have discussions and just a fun time in the classroom. So I welcome any questions you have right now, and feel free to type it in the chat, or you can unmute. I think we have small enough group where we can do that as well.
Thank you, Chris, and thank you everyone for attending the session. Please, like Chris said, if you have additional questions, go ahead and type them in the chat. Looks like Christina says, this is very interesting. I teach my forensics middle school students about DNA. They do strawberry DNA extraction lab. So she'd love to take this and simplify it.
Yeah, absolutely. This is a great one. And I don't know if, with forensics, I also sometimes teach forensic science. And I talk about with inheritance of traits like X and Y chromosome inheritance, and with Y chromosome, with the Y chromosome, you can do the YSTR analysis and track male paternal lineage, especially as they're looking at, when we're looking at samples, forensic samples, and then on a different note, mitochondrial DNA. So you could talk about the differences between nuclear DNA versus mitochondrial DNA as well.
Perfect. Thank you, Chris. That's awesome.
Yeah. Any other questions? Are there any folks out there who've actually have these kits, have used them before, have any sort of hints or tips?
All right, well, I want to give you a couple of reminders that Chris used today, the Chromosome Connection Kit and the Chromosome Student Modeling Pack. So make sure you have completed the free Student Modeling Pack registration link. Heather should have posted in the chat. And please use code CASE21 for 20% off most of our products. And then you will be sent a session evaluation afterwards. So please fill that out. That helps us in determining what types of sessions to present. Again, if you would like to have a little discussion, we'll stay on. We've got about 5 more minutes, and you can unmute yourself. Also, click on that link that Heather put in the chat earlier about the CBM summer courses. Tim yesterday did a session on CRISPR, and he said that they'll be doing a webinar. And so if you click on that link, there should be an announcement later on about when that it's a 5-session webinar series on CRISPR-Cas9. And we will stay on if anyone has any additional questions or thoughts.
Fantastic. Christina, thank you for your feedback there. Definitely, I love to use any models I can in my classroom as well. Yeah, sounds like you do as well. Oh, and Ella, I appreciate you with the tip that you got to model along with us at home. So fantastic. I'm so glad that you pre-registered and could participate too.
I think, Lizette, that you could probably stop the recording now.